Yeah. Hello, everyone. Good evening. So, any doubt in the microarray pipeline or in the sorting of significant genes and the upregulated, downregulated? Anything you want to know?
Ma'am,
Mm-hmm.
the test which you gave no last time,
Okay.
so I was not able to access that day because it was lot of network issues. So I was not able to load anything. Not even my Google Meet was working correctly. So can you share the link so that I can?
Ma'am, that thing you have to ask from the HR, ma'am.
Okay, okay.
Yeah. Okay. So, everyone is having the upregulated and the downregulated genes for the treated one versus control.
Yes, ma'am.
Okay, so in today's
Yes,
session, we are going to see the enrichment analysis. So, enrichment analysis, we will see first thing is the pathway analysis. Okay, so for that, we are going to use one database that is the KEG mapper. So, here we have to provide the IDs. Okay, so what you guys can do, this is my upregulated genes, and from this, you can copy the gene symbol in the next sheet. So this is my upregulated gene. So I want to highlight my upregulated genes and downregulated genes in the pathway also. Okay. So I will just write here red and then just autofill it. Now for the downregulated genes also, just copy the gene symbol and below just paste your downregulated genes. So in the KEG database, already the genes are present and they are highlighted with the green color. Okay. So that means if I provide a green color to this my data, I cannot differentiate my specific genes, okay, from the KEG database. So what I will do here, I'm going to highlight my down-regulated genes with the help of blue color. Then what you can do, just again autofill it. Then simply copy all the gene symbol and paste here in this box. So if you remember, this data has been taken from human, so that is why you can just select the HSA. So HSA is code for the human. So what if you are having a different organism? That time you cannot select this HSA because it is not going to take this data. So what you can do, just generate the code from here. And for example, I want to work on cow, just type here cow and you will get a code here, specific code for the organism. You can use here BTA code, that is for specifically cow, right? So this is how you can generate the code and then you can execute this. So after putting all the genes, you can see total 335 pathways are involved in your data. And this HSA is your just a specific code for your pathways, and this is my pathway name, and this is just a specific organism they are listing here, that is a human. And 130, 49, the numbers which is highlighted in the brackets, these are the number of genes that are present from our data. Okay, so for example, I want to work on this Huntington disease. Just select the, sorry, yeah, just select this particular code, and then you can see our genes are highlighted with the red and blue color, and the genes that are present already there in the KEGG database, that you can see the green color here, right? So that is why I've taken the blue color for my down regulated genes. So you can clearly see the difference between the up regulated and the down regulated genes. And now if you want to download this image, you can download from this image here. And if you want to study more regarding this particular pathway, you can just select this show description or go to the pathway entry. You will get the entire information. This is the pathway name. This is the description they are giving you. Then again the image, and you can see the gene names also. Whatever the genes that are present in your pathway, that gene names you can see here along with that. Okay. Next, what if I want to save this data? Okay, because every time I cannot open the Excel, and then we cannot just run it again and again in the KEGG database. So for the future, what you can do, just simply copy all the IDs, and then go to new sheet, and then you can paste here. So are you doing along with me? Any doubt till here? And you can rename this pathway analysis. So everyone pasted this data in the
Yes,
Excel.
ma'am.
Amit sir, Shri Charan sir.
Yes.
Okay. So what you can do, like just select the A column, again go to data, then we can sort. Why? Because you can see there is a gap in between, right? So you can sort on the basis of A to Z here, column A. Just make it okay. Done.
Yes, ma'am.
Now you can see this thing, like this human is common in each and every like pathway, right? So what you can do, just simply copy from here, from this hyphen until this bracket, then just select a column and type control H here, and you can replace with this hashtag. Done. Now I want to separate the gene and also the pathway name. So for that, what you have to do, select the A column, go to text to column, then delimited, next, go to other, put here hashtag, then finish. Then insert one row here, and you can write here pathway name. Next to that, you can see the number of genes involved in the pathway. And then just select the B column. Again, go to the data, or you can just type Control H. And you can replace single bracket, replace all.
Ma'am, please repeat.
Which sir? From
That B column.
this B column.
Yeah, yes,
Uh, so like this data you are having.
yes.
So just select the B column, then
Okay.
Control H, and put this single bracket here, sir. Then replace all.
Done.
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 Okay. So what if you are working on a particular like pathway, and so for that, what you can do, just like open any one of the pathway. So for example, you are working on this, you can just download the image, and you can also insert the image here in the Excel sheet. Go to insert pictures, this device, like this you can insert. Okay. Any doubt here in the KEGG database, in the pathway analysis?
Can you show once more how to insert?
Okay, okay. Ma'am, go to insert here, pictures, this device, then go to downloads, and in the downloads you will get the image, then you can insert. Because in real time we are going to work on only on particular like specific pathway, right? So for that purpose I told you like how to insert the picture, so that you can have like one data in the one Excel sheet only, right? So every time we cannot go back to the SCAG database, and we cannot just analyze the data from there. So that is why how to secure this data you can do like this. Any doubt till here in the pathway analysis? Sanya ma'am, Namata ma'am, Chiricharan sir.
No, ma'am, it's clear.
Next, we are going to do the functional enrichment. So that we are going to do with the help of one database, that is the Shiny Geo. Just open this database. So here we are going to paste the gene names, or you can put in sample ID, symbol, gene ID. You can see here. So, for example, for this particular treated one versus control, I am working on the down regulated genes. So it's up to you which data you are working. So that data you can just simply copy and do the analysis. Okay. So for example, okay. So for this down regulated genes, simply copy the gene symbol. If you want to work on significant genes, total significant, you can just put the symbols here. Yeah, submit this data. Pasted. So it's up to you how many pathways you want. What if you want 30 pathways? Just submit this 30 and then it will list you the data again. So yeah, here we can see first thing the enrichment table. So what is this fold enrichment? Can anyone tell me? What is the fold enrichment?
How much folds the gene is upregulated or downregulated?
Okay. So if you want to study regarding this pathway enrichment, what is the p-value? You can just study the n genes is the number of genes in the pathway that is overlapping with our gene list. So the pathway genes is total number of genes in a pathway, and the enrichment, as you said, it will give you the magnitude of the enrichment. So higher value will indicate the more enrichment here. So this value will significance how much more frequently genes are associated with that particular pathway. Okay. So that it is going to tell you here. So you can see it this the down regulated genes, these are involved in this particular pathways, arginine, the mRNA, along with that fold enrichment values they are giving, and the number of genes that are involved. You can see here. So n genes is what? The number of genes in that pathway that is overlapping with our gene list. So this is the genes that are coming from our gene list, okay, from our data. And this is the genes that are already there in the shiny geo. Got it? The difference between n genes and the pathway genes.
Yes, ma'am.
So this is the chart here. You can see this is the lollipop chart. If you want to change your chart to the bar plot, you can also do that. You can see like this. Then dot plot maybe. So what is this plot is indicating? Just wait. Yeah. Can you see this figure properly now? So this x-axis is displaying what? The name of different biological pathway, which is representing a specific pathway. You can see the name of the pathway on this y-axis. And next, you can see the fold enrichment. The length of each bar is indicating the fold enrichment of that pathway. So this value is signifying again the how frequently the gene is associated with this particular pathway. Okay. So here you can see they are like creating the plot with the help of minus log 10 value, that is the FDR here. So the color of this bar is encoding the significance of the enrichment, which is represented by this minus log 10. So so that means the higher the log 10 FDR value, indicating a more significant enrichment, which will suggest that the observed enrichment is likely less be due to the chance here. Okay. So here you can see, can you tell me the which is having the highest enrichment here? Can you tell me? Shri Charan sir?
Argin, arginine biosynthesis.
This one, Shri
mRNA
Charan sir,
surveillance pathway.
Amit sir,
mRNA surveillance pathway.
this one. So you can see from this table also, I told you the fold enrichment, if it is higher, that means it is indicating what? The genes is significant and it is showing the enrichment which is likely due to be less chance. You can see from here the fold enrichment of the arginine, as ma'am said, it is correct. The arginine is having the largest fold change. Can you see the 6.6 here? That is why the length of this plot of this bar, you can see the it is higher. Can you see that? Clear this plot?
Yes, ma'am.
Okay. Can you tell me regarding this tree now? What this figure is indicating? Any idea?
How they are connected.
Yes, ma'am. So actually this figure is appeared to be a dendrogram, a tree-like diagram which is used to visualize the relationship between the data point. So in this case, the data points are likely to the biological pathways. You can see here, and you can also update from here. Okay, we will do later on. So by looking at this, you can see they have formed the cluster also. And there are some vertical lines and the label also. So this vertical lines, each vertical line, it is representing a branch in the dendrogram. And the length of the branch indicate the distance or the similarity between the data points it connect. And the label you can see here, that means the label on the branch, it is corresponding to specific pathway. And the numbers, sometime you are getting the numbers also. Can you see numbers here next to the labels here? So which is likely to be representing some statistical measure. Possibly it can be a p-value or the fold change. Okay. So by looking at this figure, the dendrogram shows some cluster of the pathway. Can you see the cluster of this pathway? This two are indicating. So this is, we call it as a cluster here. These are the vertical branches, which is indicating the specific pathway. And these two cluster you can see here, it is showing some similarity between each other. That is why they are forming some cluster in this dendrogram. Okay. Clear? And what is the statistical score here? So this is the number which is indicating some statistical significance of the association between the pathway, and the higher the value, it will suggest more the stronger association. Remember that. Clear? Done? So for example, we are going to do the functional enrichment analysis. So functional enrichment is what? Like we are going to see the biological process, cellular component, and the molecular function. So what you can do, just select the geobiological process. So these are some biological pathway, and these are number of genes from our list that are involved in this pathways. Okay, so what you guys can do, just download this. And we can just label this as the biological process. Make one folder here inside the microarray. And you can type here functional enrichment. Rename this file, that is the biological process. Like this, you can also download from for the cellular component, then molecular function. So these three functions you are going to download, and you can download from here the table, and you can put inside this folder. Downloaded.
Yes, ma'am.
Shri Charan sir, Amit sir, Sanya ma'am. Namrata ma'am, did you download it for cellular molecular?
It is getting down.
Okay, okay. You can download.
Done.
Okay.
Ma'am, I have network issue. Can you please repeat?
Uh, okay.
From biological process.
Okay. Mm, just wait, sir. Can you see my screen, sir?
Yes.
Actually, in real time, like whatever the data you want to study, so on that like specific reason you are going to analyze here. Okay, right now I have just put here down regulated genes. What if you are going to work on significant gene? You can load entire significant genes here. Okay, in the
Okay.
shiny GU. So for example, you are doing any functional enrichment analysis, so you can see it is divided into three functions. You can see biological, cellular, and the molecular. So one by one you can just select this biological process. It will be updated to the biological pathway. Okay, so these are some biological pathway here, and these are number of genes that are involved in this particular pathway, and along with that the fold enrichment also you are getting here. Okay, so you can just download the result, and then that particular file you can rename it as the biological process. Then likewise you have to download for cellular component and the molecular function. Just download these three files.
Done.
Okay. So all of you, what about you? Done, Namata ma'am? Okay, you can open this biological process. You can see along for this particular pathway, that is the macromolecular modification, you are getting the genes also. So these are genes which are coming from our data. And now if you want to sort this data on the basis of the fold enrichment, because I want to see like which function is highly enriched in this biological process, so what you can do, just put the filter here. Mhm. Okay, so it is giving you decimal number. So you can sort on the basis of this largest to smallest. Okay, now after putting this filter, you can see the pathway name. So from this particular data, can we say in the biological process, the endoplasmic reticulum tubular network formation is highly enriched in our data. Okay. Next, the second pathway is this, and then third, you can just do this thing similar for the cellular component and the molecular also, along with the gene names you can see. Can you just sort like in the cellular also and the molecular also?
Ma'am, here we have taken the down-regulated genes.
Yes, ma'am.
So would it mean that the genes are down-regulated by this much?
Yes, ma'am, right. Because we have taken in this data that is the down regulated genes, right? So, as you said, it is correct, ma'am. So, down regulated genes are highly enriched here in this particular pathway, ma'am. Okay. Actually, in the functional enrichment analysis, what we are doing, like we want to know which function is highly enriched for our specific genes. So, that we are going to see here in this enrichment analysis. So meanwhile, who is not having the cytoscape in their system, they can download Shirchansan, are you having this cytoscape?
No, ma'am.
Namata ma'am, Amit sir.
No.
Yes, ma'am. I have.
Okay. So just browse it, go to the download cytoscape, and then go to the older version. Just select on this version 3.8.2. And if your system is Windows, you can go with the window here. If your system is Mac, then Mac. So for the window, if your processor is 64, you can just install it directly from this window 64bit.exe. So I guess everyone is having window, you can install it, this tool. So with the help of this cytoscape, we are going to see the network analysis. And then after network analysis, we'll see the heat map and the volcano plot in coming session. So Shri Charan sir and Namrata ma'am, if you're not having, you can download this.
Okay, ma'am.
Yes. And also we can create the chart for this functional enrichment also. So for example, uh, we are going to delete this URL ID and the genes from here and the pathways also. And this enrichment value. So what if I want to create a pie chart for this particular 10? Okay. So what I will do, I will just insert one row here and I'm going to select this particular data. Then go to insert. Maybe you can generate with the help of 3D. Why it is stuck? Yeah. Can you see this pie chart? Now on this pie chart, just right click it, go to add data labels, and you will see different colors I'll highlighted, which is showing the specific pathways. And you can see this numbers, what it is indicating the number of genes that are involved. Okay. Clear? Any doubt in this pie chart? You can also try. Done.
Yes, ma'am.
Okay. So you can also use one other database for the functional enrichment. You can try this also, Panther. Okay, just try it at your home. So by this database also you can do the function enrichment. So same thing, you will get the molecular function also, cellular component also, and the biological process also. Okay, sometime what happen in the geo or maybe in this Panther, you are not getting the entire information. You cannot rely on particular one database, right? So that time you can use the different databases for the functional enrichment also. The purpose is same. That same thing you will get there also, the number of genes, then the pathways that are involved in the biological process, along with the number of genes you will get there also in the Panther database. But you should know how to operate that database, okay? And how to create this pie chart in the Excel. So did you got it how to create this and what we did in the function enrichment today? Any doubt in the pathway and the function enrichment? Because from tomorrow onwards we are going to see the network analysis with the help of two database, that is the string database and the cytoscape. Okay. And make sure like everyone is having tomorrow the cytoscape tool in their system. Any doubt, Shri Charan sir, Ram sir, who's Amit sir,
No, ma'am.
Sonali ma'am, is it okay? Did you got this?
Yes, ma'am.
Okay, Namata ma'am,
Got it.
okay, Sanya ma'am. Okay, then we can end it here, and you can try this function enrichment with the help of Panther database. If you have any doubt, just let me know tomorrow also. Okay, then we'll move ahead with the network analysis tomorrow.
Okay, ma'am.
Okay then. Okay then. Bye bye.
Thank you, ma'am.
Yeah.
Thank you.